All right, everyone, welcome back. We are now starting block 3. We're going to start looking at the cardiovascular system, which will include a little bit of the lymphatic system. So we have here chapters 18 and 19, but it's also going to include a little bit of chapter 20. And when we go through this, we're going to be jumping around a little bit compared to how it's presented in the textbook, because it will make a little bit more sense the order that we cover it in. So really what we're going to do is we're going to begin with a little bit of chapter 19. Then we are going to do a little bit of chapter 20. We're going to fall back and go through pretty much all of chapter 18. And then we're going to jump ahead again and wrap up with just a little bit of chapter 19. Now, when you look at chapter 19 in your textbook, there is a lot of coverage of the different named blood vessels. We are not going to go through that. We did some of it in lab already, but most of that we're just going to skip over. We are not concerned with naming blood vessels other than the few that I will specifically talk about in some of these lectures. But most of chapter 19 we can skip over. Only be concerned with the parts of it that you see in your outline and here in these lectures. So as always, let's start with our not really attendance questions. First, what is the primary function of albumin? And next, if there is greater osmotic pressure on one side of a membrane than on the other, what will be the result? So go ahead and pause your video, try to answer those. Hopefully you've done that, and these are going to be things that come into discussion in today's lecture. First, what is the primary function of albumin? And this is albumin with an I-N, not an E-N. Albumin with an E-N is egg white. That's not what we're talking about. We're talking about albumin with an I-N, and that is the most abundant plasma protein. So think back to when we were talking about plasma. Well, albumin is the primary contributor to osmotic pressure in the blood or in the plasma. Remember, osmotic pressure, it's pulling liquid towards itself. It is opposing hydrostatic pressure. Or another way that we commonly say that in my class is it is a solute and solutes suck. They suck water towards themselves. So that brings us to the next question. If there is a greater osmotic or pulling pressure on one side of a membrane than on the other, what will be the result? Well, liquid, probably water, but the liquid portion is going to move towards the area where there is greater osmotic pressure. So from the side of the membrane with the lower osmotic pressure, water or liquid will cross that membrane towards the side with the greater osmotic pressure, assuming that membrane is permeable to that liquid. So now we're going to talk today about blood vessels. And there are different types of blood vessels in the body, all of them with different names. We are not, again, going to be concerned with the names of the majority of them. We are going to be learning about what do those blood vessels do. And the first blood vessel type we're going to talk about are arteries. Arteries are the blood vessels that take blood away from the heart. We'll come back to that in just a moment. Arteries tend to be pretty large vessels. There are small arteries, but in general, arteries are pretty large vessels. And when we look at them, if we took a cross section, we would see that there are layers to those arteries. There are 3 layers. And if we go from the inside out, the hollow part of an artery, and we don't see the word here, but the hollow part of an artery where the blood is found is called the lumen. And really, any hollow organ, the hollow part of it is going to be the lumen. Now, the inside wall of an artery, that layer is called the tunica intima. Tunica just means layer. So the tunica intima is the layer on the inside of a blood vessel, or an artery in this case, and this is what the blood would actually come into contact with. And it is made of simple squamous epithelium. Now, moving outward, the next layer, which is the middle layer, is called the tunica media. And the tunica media is primarily smooth muscle. There are some elastic fibers in there to give it that ability to stretch and snap back. But primarily smooth muscle, and smooth muscle, remember, just like other muscles, it can contract and relax, and this regulates the diameter of the blood vessel, which is called vasodiameter. So the diameter of a blood vessel is called the vasodiameter, and it is regulated by the smooth muscle of the tunica media. Now the outermost layer, so if you were to open someone up and see their blood vessels intact, the part of the blood vessel that you would see, the outside, is called the tunica adventitia. Sometimes you will also see it called the tunica externa. These are two different names for the same thing. The tunica adventitia, the tunica externa, that is the outside layer of the blood vessel, and it is primarily made of collagen fibers. Remember, collagen fibers, very tough connective tissue fibers. And these are just for protection, for durability. So, vasodiameter, we said, was the diameter of a blood vessel. And the vasodiameter is regulated by that smooth muscle of the tunica media. If that muscle relaxes, then the vasodiameter will be larger. If the smooth muscle contracts, then the vasodiameter will be smaller. Now, vasodiameter is controlled by the sympathetic nervous system. When sympathetic nervous system activity is up, or when sympathetic nervous system activity is down, that is going to alter the vasodiameter. Now notice there's nothing here about the parasympathetic nervous system. As a general rule, the sympathetic nervous system and the parasympathetic nervous system control those parts of our body that we don't consciously think about. But blood vessels do not have any input from the parasympathetic nervous system. It is entirely under the control of the sympathetic nervous system. And if sympathetic nervous system goes up, we get vasoconstriction. The vasodiameter gets smaller. If the sympathetic nervous system activity is down, we get vasodilation, and the vasodiameter gets bigger. So, sympathetic nervous system active, vasoconstriction. Sympathetic nervous system deactivated, we get vasodilation. Now let's think about what happens when we get vasoconstriction and vasodilation. If we constrict a blood vessel, if we make the vasodiameter smaller, is the blood inside going to be under greater pressure or lesser pressure? If we squeeze the vessel smaller, if we vasoconstrict, we are squeezing on the blood that's inside of it. That blood now is under greater pressure. So let's put all of that together. When sympathetic nervous system is activated, when it is elevated or up, we get vasoconstriction. When we get vasoconstriction, the vasodiameter gets smaller and blood pressure goes up. And the opposite is true. When sympathetic nervous system is deactivated, when it is down, we get vasodilation and the vasodiameter gets bigger. We squeeze less on the blood that's inside. So blood pressure goes down. Now, as I just said a moment ago, the function of arteries is to carry blood away from the heart. We carry blood away from the heart. Now, with that said, I'm going to tell you a question that will be on every single exam going forward. It will be on your cardiovascular exam. It will be on the Block 4 exam, the Block 5 exam, and the final exam. The question is going to be, which of the following is true? Or it will probably be worded as arteries blank. And the responses will be carry blood towards the heart, carry blood away from the heart, always carry oxygenated blood, always carry deoxygenated blood, or more than one of the above. So, tell me about arteries. What would be the correct answer? Would it be that they carry blood away from the heart, carry blood towards the heart, always carry oxygenated blood, always carry deoxygenated blood, or more than one of the above? So, not only is that question going to be on every exam going forward, I'm going to tell you the answer. And I always do this, but without fail, students will still miss this question on the exam, even though I'm telling you what the wording of the question is, what the answer of the question is going to be. There will still be students that miss it because they have misconceptions about arteries. The correct answer is arteries carry blood away from the heart. There is no relationship between the oxygen in the blood and arteries. Yes, most of the arteries in the body carry oxygenated blood, but not all of them. Some arteries go away from the heart, going towards the lungs to pick oxygen up. Those arteries are low in oxygen. They are deoxygenated. So, when you see the question, arteries blank, the correct answer is they carry blood away from the heart. Make sure to remember that. So as we go away from the heart, the vessels are going to branch and branch and branch. And every time they branch, they get a little bit smaller. So leaving the heart, we go into the arteries, and then smaller arteries and smaller arteries, until eventually the arteries are so small that they are a little bit differently structured. And we call these arterioles. Arterioles really are just very, very small arteries. But we also will sometimes see them referred to as resistance vessels. And we call them resistance vessels because they regulate peripheral resistance, abbreviated PR. Now, we are going to learn a lot of terms in this chapter, or these chapters. So far we have learned about vasodiameter, vasoconstriction, and vasodilation. Now we are going to talk about peripheral resistance, PR. Peripheral resistance is the resistance provided by the vessels against the blood. That's the technical definition. I like to say peripheral resistance is really how easy is it for blood to move through this vessel. And several different things regulate peripheral resistance or influence peripheral resistance. Going back to what we just learned, vasodiameter influences peripheral resistance. If a vessel is very, very, very constricted, peripheral resistance will be up. If a vessel is very, very, very dilated, peripheral resistance will be down. So one of the things that regulates peripheral resistance is vasodiameter. And arterioles regulate peripheral resistance by constricting or dilating. So this, in turn, significantly affects blood pressure. As peripheral resistance goes up, it's harder for blood to move through the vessel, so blood pressure goes up. if peripheral resistance goes down, it's easier for blood to move through the vessel, so blood pressure goes down. So PR and BP, peripheral resistance and blood pressure, have an equal relationship, meaning when one goes up, the other goes up. When one goes down, the other goes down. But more specifically, PR happens and BP results. So if PR goes up, BP goes up. If PR goes down, BP goes down. They are related directly. Whatever PR does, blood pressure does the same thing. Now, as blood moves through arterioles, it's going to flow into capillaries. Capillaries are so small, they are microscopic. They are so microscopic that as blood flows through, even the blood cells have to kind of squeeze their way through because they are smaller around or smaller in diameter than erythrocytes, red blood cells. Except for sperm cells, erythrocytes are the smallest cells in the body. So these capillaries, these very small blood vessels, are so small that even the smallest cells in the body cannot fit through. They are only a single layer thick, and that layer is the tunica intima. Remember, the tunica intima is simple squamous epithelium. So one very, very thin layer, which is important because the function of capillaries, this is where fluid exchange takes place. Stuff that's in the blood needs to be able to get out into the tissues to supply the tissues with oxygen and nutrients. And waste and carbon dioxide from the tissues needs to be able to get into the capillary to be carried away. Simple squamous epithelium, think back to Bio 137. What was the function? It made diffusion very, very easy. So capillaries are built so that it is really, really easy for that fluid exchange to take place. Now, capillaries, they're found all over the body, except they're not really found in tendons, ligaments, and cartilage, which is why if you ever damage a tendon or a ligament or a cartilage, it can take a really long time to heal, if it heals at all, because we need a good blood flow for healing to take place. So here's our capillary. Capillaries, they're really permeable, which again, we need them to be so that things can get in and out. Here is that single layer, that simple squamous epithelium, that tunica intima. And there is a wrapping around it. It's kind of not really a part of the capillary. This is that basement membrane that epithelial tissue is attached to. So this is not a part of the capillary, but it's around the capillary. Inside, remember, this is the lumen. Here we see it blown up a little bit down here. So this is the lumen. And then we have the wall of the capillary. Now, depending on what substance it is that's moving in and out of this capillary, it can move in and out a few different ways. One way is by passing in between two adjacent cells through something called an intracellular cleft. Capillaries, the cells are not joined very tightly together because we want stuff to move in and out of a capillary. Things can just diffuse straight across the wall of the capillary. Or in some cases, we have fenestrations, which are basically holes in the wall of the capillary. Not all capillaries have them, but some do. And if it's a really big substance, or if it's quite a bit of water, well, we have vesicular transport. Something moves into the capillary wall on one side, travels through in a vesicle, and then exits on the other side. So what does the capillary bed look like? Because when we have capillaries, it's not a single, very small vessel. Instead, it's more like a spider web, kind of like what we see here. The red over on this side is the arteriole, bringing blood, again, away from the heart, going towards the tissue. Here is the capillary bed. Blood branches out and moves through all of these different vessels, and then it leaves through the capillary bed into this blue side and will eventually be carried back to the heart. But sometimes a tissue has very high demand, so it needs a lot of oxygen, it needs a lot of nutrients, it's building up a lot of waste to be carried away. So blood will move into all of these different things, these different branches that we call true capillaries. And more and more and more blood flowing into these capillaries, these true capillaries, means that we have more of that exchange taking place. More stuff moving out of the capillary into the tissue, more stuff moving out of the tissue into the capillary, and then blood flows on through. But sometimes tissue doesn't have a very high demand. We don't need a lot of nutrients and oxygen. We don't build up a lot of waste. So there's no reason to send blood into these. So instead, blood comes in and passes pretty much straight through the middle, bypassing it all and leaving the other side. So let's see the difference in the two and how it's regulated. Well, if a tissue does not have a high demand, then what happens is blood comes in through this arteriole and goes into something called a meta-arteriole. And then it moves into something called a thoroughfare channel. And then it exits the other side to be carried away back to the heart. This bypasses the true capillaries. This is a way to just quickly get blood through without wasting it out into these other pathways. That meta-arteriole and this thoroughfare channel together is called a vascular shunt. Shunt just means to quickly move something through easily. So we are moving through a vascular shunt without going into the true capillaries. This happens when tissue does not have a high demand. And it's regulated by these very, very small rings of smooth muscle. These are called precapillary sphincters. A sphincter is just a ring of smooth muscle. So these precapillary sphincters would constrict, preventing blood from moving out into these true capillaries. But if the tissue has a high demand, if it needs a lot of nutrients and oxygen, well, those precapillary sphincters dilate and blood floods into those true capillaries. That tissue is basically flooded with blood entering these true capillaries. This allows a lot of nutrients and oxygen to be dropped off. It allows a lot of waste and carbon dioxide to be picked up. So here we see a capillary bed. Now, normally there's about 10 to 100 of these paths in one capillary bed, which also means there's about 10 to 100 of these vascular shunts through one capillary bed. But how do we know when there's going to be a high demand or not? Let's take a look at the difference between high and low demand. Let's say you have just eaten a really big dinner and now you are relaxing for the evening on the couch watching TV. Your skeletal muscles, the muscles that move you around when you're being active, they're kind of relaxed. They're not needing a lot of oxygen. They're not needing a lot of nutrients. So if we looked at the capillaries in those skeletal muscles, let's say in your legs, they would look like this bottom image down here. Those precapillary sphincters would be constricted and blood would be getting shunted through this vascular shunt, bypassing most of those skeletal muscles. But if we looked at the blood vessels around your stomach and around your intestines, well, we've just eaten a meal and now we're needing to absorb a lot of those nutrients. That would look more like this top image here. If we looked around your intestines, all of the capillaries surrounding your intestines, those precapillary sphincters would be dilated to allow blood into those capillaries. So your intestines would be getting flooded with blood into all of these true capillaries, absorbing all those nutrients that we just ate. But now a few hours later, you're feeling good, you decide it's time to go for a run. Now we're not so interested in getting nutrients from our intestines. We've already absorbed most of those. So these precapillary sphincters around your digestive system are going to constrict. Not as much blood is going to be going to your digestive system capillaries. They're going to look more like what we see down here at the bottom. Now blood is going to get shunted through, kind of bypassing your digestive system. Meanwhile, those capillaries around your muscles in your leg, as you start to go for your jog, well, those precapillary sphincters are going to dilate, allowing blood to rush in to those true capillaries around your muscles, bringing them lots of nutrients, bringing them lots of oxygen, carrying away carbon dioxide, carrying away all of the waste that's building up. So these go back and forth from one to the other based on tissue demand. But let's go back to this image real quick. As blood is coming out of a capillary, now it's going to go back towards the heart. So we enter this next vessel. This is called a venule. And venules are just very, very small veins. But there's different types of venules, so they have different makeup. As soon as we exit a capillary, we're in something really called a post-capillary venule. But these are very similar to capillaries. They are simple squamous epithelium. They're one layer thick. They are very porous. And the reason they are porous is because those leukocytes that we've learned about previously, this is how they get out of your bloodstream. Remember, things like monocytes leave your bloodstream to go out into the tissue as macrophages. This is one of the easy spots for them to get out. Now, some of them also have a thin tunica media and a thin tunica externa, so they do have a little bit thicker walls. But then as they get closer and closer and closer to the heart, they're going to join together, making small veins join together to make bigger veins until eventually we get to some of the really big veins. Now veins, they have the same layers as arteries. But the difference in the wall of a vein and the wall of an artery is that a vein will have a much thinner tunica media. That is because as we exit the heart, arteries are getting blood pushed into them from the heart. Arteries have a lot of pressure inside of them, so they need very thick walls. And as a result, their lumen is actually pretty small. Veins, on the other hand, we're very far away from the heart. We're on our way back towards the heart, actually. So as we come out of capillaries into venules and veins, the pressure is very low. And because the pressure is very low, we do not need really thick walls. The tunica media is extremely thin. But as a result, that means the lumen of a vein is very large compared to an artery. And since the pressure is so low, blood runs the risk of going back the wrong way. Think of blood in your foot. For it to get back to your heart, it has to fight against gravity. But since pressure is so low, it's hard for it to fight against gravity. So we have valves in our veins, and those valves are one way. They let blood go through going towards the heart, but it prevents blood from going back away from the heart. So as your heart beats and pressure builds, blood is pushed through. As your heart relaxes and pressure drops, blood is prevented from dropping back down towards the earth, towards gravity, because of those valves. So what's the function of a vein? Exactly the opposite of the function of an artery. Veins carry blood towards the heart. And like arteries, nothing to do with oxygen. Yes, most veins are deoxygenated, but not all of them. Veins coming back away from your lungs going towards your heart, they carry a lot of oxygen. They just picked a lot of oxygen up at the lungs. So the function of a vein is to carry blood towards the heart. Veins are sometimes referred to as capacitance vessels. Because they have that large lumen where the blood is found, they can contain up to 65% of the blood in your body at any moment. And like I said, the further you get away from the heart, the lower the pressure. Blood in your veins only has about 5 to 7 millimeters of mercury of pressure. So a couple of things to point out here. One, there's a large vessel that empties into the right side of your heart called the vena cava. Sometimes its pressure is so low it's not really measurable. And the other thing is mmHg, millimeters of mercury. A few times this semester, actually several times this semester, we are going to be talking about different types of pressure. Every time we talk about pressure in this class, whether it's blood pressure, whether it's atmospheric pressure, whatever it is, when we talk about pressure, we are always going to use the unit mmHg, millimeters of mercury. You do need to know that that means millimeters of mercury, and anytime that you have to write a pressure, make sure you include the units mmHg after it. So since veins have such low pressure, they depend on more than just the heart to move blood through them. We have muscular pumps. Well, think of the blood vessels in your leg. As you move around, your muscles in your leg are going to squeeze and relax and squeeze and relax, and the veins move kind of in between these muscles. So every time your muscles contract and relax, they get fatter and thinner, and it's kind of like they are squeezing or milking your veins. They're squeezing blood through. The same thing in our chest area. We have pulmonary pumps. Every time you take a breath, your lungs increase in size and then decrease in size, and the pressure inside your chest wall increases and decreases, and these also squeeze on those veins, helping to squeeze blood through. And here's a way to visualize everything that we just talked about. Here's a blood vessel. Let's say it's coming up from your leg, so blood flow is going from the bottom of this image towards the top of the image. And what we see is these valves inside. These valves are shaped so that blood can go from the bottom towards the top in this image. And here we see the muscles. And every time these muscles contract and relax, they squeeze on this blood vessel, this vein. And as they squeeze, blood can go up towards the heart, but it cannot go down because this valve is closed. So the valves allow one-way blood flow in veins. Arteries don't need that because arteries have a lot of high pressure, so they don't need these valves. Veins, though, because their pressure is so low, they do. So here's a microscopic side-by-side comparison of an artery and a vein. Now, if we were to fully inflate both of these, they would be about the same outside diameter. But up here at the top, this is an artery. Here is that tunica adventitia or the tunica externa. Here is that really, really thick tunica media where that smooth muscle is. This clear area here is the lumen where the blood would be found. Down here is a vein that would have the same external diameter. We can see there's not very much of that tunica media at all. But look how big the lumen is. So these are about the same outside diameter, but you can see the difference in the thickness of that tunica media, and you can see the difference in the size of the lumen. So let's talk about something that can go wrong with blood vessels. Here we're going to talk about varicose veins. Varicose veins are something that happen to most people at some point, but they are aggravated, they are made more likely if you are either on your feet a lot, if you are overweight, or if your job requires you to do a lot of carrying of weights. If you are pregnant or have had a baby, if you do a lot of squatting and standing, those kind of things will result in varicose veins being much more likely. So here's how varicose veins happen. We are looking at your leg, and varicose veins tend to happen in legs, but they can happen elsewhere. So we're just looking at a leg here. So here is a normal vein over on the left. Blood moves upward through the vein. It cannot go backward, back down because of those valves. But in any of those situations like I mentioned, what happens is blood goes up, but let's say you do a lot of just standing. Well, blood is constantly trying to be pulled back downward because of gravity. Because the walls of veins are very thin, and really the valves are just an extension of the wall. They are also simple squamous epithelium. Well, what's going to happen is those veins cannot withstand the force of gravity for extended periods of time. The blood gets through the veins. We say that the veins, through the valves, we say that the valves are incompetent. They are unable to do their job. So blood goes back down. Now we have more blood than there should be inside this part of the vein, in between this valve and the next. More blood has come back. And since the walls are really, really thin, they distend. They bulge outward. And that's what varicose veins are. It's when blood goes back the wrong way, there's too much pressure in one area, and the walls bulge outward. Now, those are varicose veins. And like I said, varicose veins, they tend to happen in the legs, but they could happen anywhere in the body, including one place where they could be extremely painful, and that's hemorrhoids. Hemorrhoids are varicose veins. They are just varicose veins around the anus. And this tends to happen with people who are either constipated regularly, or if they have delivered a baby or carried a baby, or anything that's causing them to squeeze their stomach muscles. And that results in varicose veins around the anus. Okay, changing gears a little bit, now we are going to be looking at capillaries and how they do their job. Remember, their job is that's where fluid exchange takes place. And I say fluid exchange because what happens at a capillary is not oxygen moves out and nutrients moves out. It is liquid, essentially plasma, not entirely plasma, but similar. Liquid moves from the capillary into the interstitium, or from the interstitium into the capillary. And in that liquid, we have dissolved oxygen, dissolved nutrients, dissolved carbon dioxide, dissolved waste. So as the capillary moves in, as the fluid moves in and out of the capillary, there are things in that fluid that are what are really moving in and out of the capillary. So here I've kind of shown a red tube. This is a capillary. On the left side of the screen, this is where the arteriole is feeding blood into the capillary. So we say this is the arteriolar end. And on the other side, this is the venular end or the venous end. Blood is moving out of the capillary into the venule to go back towards the heart. So blood is moving from the left side towards the right side. This red arrow shows us the direction of blood movement. Now this capillary is surrounded by tissue called the interstitium. The interstitium is really just, we can kind of think of it as interchangeable for tissue, but really this is the area outside of cells, because as stuff moves out of a capillary, it does not move directly into a cell. It moves to the extracellular region of the tissue. That's the interstitium. Now there are cells out here, so fluid is really moving into the interstitium and then into the cells, or it's moving from the cells into the interstitium into the capillary. So let's learn some terms now. Looking at the arteriolar end of a blood vessel, of a capillary, there's going to be blood pressing against the walls of the capillary. We could say it is liquid pressing against the walls of its container, trying to get out. By definition, remember that's what hydrostatic pressure is. Hydrostatic pressure is the pressure of a liquid against the walls of its container. If the container, in this case the walls of the capillary, if the container is permeable to the liquid, then the liquid might push all the way through. So here the hydrostatic pressure inside the capillary, so we call it the hydrostatic pressure of the capillary, HPC, hydrostatic pressure of the capillary. Sometimes you might also see this called hydrostatic pressure of the colloid. Colloid is just the plasma and the stuff dissolved in it. But I will refer to it as hydrostatic pressure of the capillary. So we have the hydrostatic pressure of the capillary pushing outward. But there's other pressures. There's something called the osmotic pressure of the capillary. The osmotic pressure of the capillary, or colloid, is all of those solutes. Primarily what? Primarily albumin. And what do those solutes do? They suck. They pull water towards themselves in all directions, including from out here in the interstitium. So they are pulling water inward. Hydrostatic pressure pushing outward, osmotic pressure pulling inward, if we're talking HPC and OPC. But there's fluid out here in the interstitium, and it is also pressing against the walls. So the hydrostatic pressure of the interstitium, or the hydrostatic pressure of the interstitial fluid, is also pressing against the walls of the capillary, but from the other side. So we have two hydrostatic pressures. One of them is pressing against the wall in this direction. One is pressing against the wall in the opposite direction. And lastly, we have the osmotic pressure of the interstitial fluid. Osmotic pressure from proteins and other solutes in the interstitium sucking water towards itself from all around the interstitium, but also from the fluid in the capillary. So now we have two hydrostatic pressures pressing against the wall of the capillary from opposite sides. We have two osmotic pressures sucking water towards themselves in opposite directions. Now that's all well and good. Those are the pressures that we're dealing with. But we need to measure them. And since they are all pressures, what unit are we going to use? mmHg, millimeters of mercury. So if we measure them down here on the end closest to the arteriole, we find that these are those values. The hydrostatic pressure of the capillary is pressing its way outward with a force of 35 millimeters of mercury. The osmotic pressure of the capillary is sucking water inward with a pressure of 26 millimeters of mercury. The hydrostatic pressure of the interstitium is so small, we just call it 0 millimeters of mercury. And that's because the liquid that's out here in the interstitium quickly moves into the cells. And lastly, the osmotic pressure of the interstitium is very, very low, and we measure it at about 1 millimeter of mercury. Now, the thing that we run into is we have two different directions of fluid movement. We have fluid moving from inside the arteriole towards the interstitium, and we have fluid moving from the interstitium into the capillary, into the blood. So, we need to do more than just have these numbers. Your book handles it a little bit differently. I think, since I don't like math, I think it's easier to do it the way that I do. So here's how I'm going to present it. We are going to say anything moving from the inside of the capillary into the interstitium, we're going to call it a positive pressure. And anything moving from the interstitium back into the capillary, into the blood, we're going to call it a negative pressure. The numbers don't change. It's just from the capillary to the interstitium is positive 35 or positive 1. From the interstitium going back in towards the blood vessel, we're going to call it negative. Negative 0, which doesn't really mean anything, and negative 26. All of those millimeters of mercury. Why does that matter? Well, we're going to take all of those numbers and add them. Positive 35 minus 26 minus 0 plus 1. We've just taken all of these and added them. And we get 10 millimeters of mercury. And since it's a positive number, that means it's going in the outward direction. It's going out of the capillary. So fluid at the arteriolar end of a capillary is moving out of the capillary into the interstitium. That is exactly what we want, because what was the capillary doing? It's delivering nutrients and oxygen. So here we are pushing fluid with nutrients and oxygen out into the interstitium so that the cells of this tissue can use it. And that's what this image in your book is showing. This is showing that pressure at the arteriolar end moving out of the capillary into the interstitium. We call that, this pressure, when we add all of them together, the net filtration pressure. Net because we've added all of them together. Filtration, remember, that was when something moves out of an artery, out of a blood vessel. So that is net filtration pressure, and it was positive 10 millimeters of mercury. Positive, so things were moving out of the capillary. But as we move through the capillary, we're losing liquid and we're getting towards the venular end. Well, at the venular end, or the venous end, we still have all of those same pressures. But some things have changed. Since we are losing liquid from the capillary, then the hydrostatic pressure of the capillary will be smaller. There's not as much liquid to push against the wall of the capillary. And this is what we get. Now, over here on the left, this is still the same thing that we just saw. But as we move through and liquid is moving out, this hydrostatic pressure of the capillary drops. Fluid is moving out here into the interstitium. By the time we get to the venular end, or the venous end of the capillary, the hydrostatic pressure is much smaller. It's only 17 millimeters of mercury instead of 35. None of the other pressures change, and that is because even though water or other liquid is moving out here into the interstitium, it's quickly moving into the cells. So that's why this is still 0. The osmotic pressure doesn't change because the proteins that are here in the plasma are too big to move out of the blood into the interstitium. So this does not change. The osmotic pressure inside the capillary also doesn't change for the same reason. Those plasma proteins that were in down here at the arteriolar end are still in down here at the venular end. Now, if we add all of those numbers up, here we get -8, 17 outward, 26 inward, 0 inward, 1 outward. We get -8 millimeters of mercury. So that tells us there is an inward pressure of 8 millimeters of mercury. At the arteriolar end, there is a net outward movement. We are supplying that tissue with nutrients and oxygen. At the venular end, fluid is moving back into the capillary. We're picking up waste and carbon dioxide to carry away from the tissue. We want fluid to leave the capillary at the arteriolar end to supply the interstitium. We want fluid to move into the capillary at the venular end to load the blood up with waste to be carried away. And that's what our image is showing here. At the venous end of a capillary, these are everything that we just drew. There is a net filtration pressure of 8 millimeters of mercury in an inward direction. But there's an issue. There is a 10 millimeter of mercury outward pressure at the arteriolar end. Fluid is being sprayed out of this capillary, essentially, with a pressure of 10 millimeters of mercury. At the venular end, we've got liquid coming back in with a pressure of 8 millimeters of mercury. What do we think the problem might be? Well, we've got more liquid leaving on the arteriolar end than we are reclaiming on the venular end. Another way to say that is more liquid leaves than we carry away. Another way to say that is we are losing liquid from our blood constantly at our capillary. Doesn't seem like much, but if this is going on at every capillary all the time, we would dehydrate in a matter of a minute or two if there wasn't a way to fix it. So we're constantly losing fluid at the capillaries, which is what we see in this image. Here's the arteriole, carries blood in, liquid leaves at the arteriolar end, liquid is reclaimed at the venous end. That's what these tiny little arrows represent. But more liquid is leaving than is regained. So we've constantly got liquid filling up out here in the interstitium. What happens to that liquid? That is the topic of our next lecture. Down here, every single capillary bed is surrounded by something called lymphatic capillaries. And those lymphatic capillaries are going to be what recovers all of that lost fluid. All right, that was a pretty long lecture today. I'm sorry for that, but it had to all fit together since it all matched. But we will pick up in our next lecture and look at these capillaries and how they work, or these lymphatic capillaries and how they work. If you have any questions, let me know. Otherwise, I'll talk to you in the next lecture.